Chapter 5, An Introduction to Carbohydrates, An Introduction to Sugars. In this chapter, we'll talk about the structure of monosaccharides, and then we'll use these building blocks to build polymers or polysaccharides. We'll talk about the major role of carbohydrates and how they're important in cell structure, cell identity, and energy storage. Carbohydrates or sugars are macromolecules, and the reason why they're so important is because they play an important role in energy, they contribute to cell structure, and they're involved in cell recognition and identity. That almost sounds like a test question, and I think it should be. So there's different types of carbohydrates. There's monosaccharides, oligosaccharides, and polysaccharides. But the truth is, I often hear the terms mono and polysaccharides, and I rarely ever hear the word oligosaccharides. So mono means one, and poly meaning many. The general formula for a carbohydrate is CH2O. Notice that's within parentheses with an N outside. So basically what it means is sugars can have a different amount of carbon, but notice because of the formula, there's always twice as many hydrogens as there are carbons and oxygens. So carbo refers to carbon and hydrate refers to water. So N can vary from 3 to over 1,000. So when we talk about a polysaccharide, we're actually talking about a very large macromolecule. So within a sugar, we can find special functional groups. And one of the functional groups is the carbonyl group, C double bond O, and I will be referring to that later on in this discussion. We can also find hydroxyl groups, OH, and then we'll talk about carbon-hydrogen bonds. Carbonyl and hydroxyl groups are polar, which indicates that they have a charge. So that means that carbohydrates are hydrophilic. Hydro meaning water, philic, water-loving. So they're attracted to water. Birds of a feather flock together. Monosaccharides are simple sugars, and it says here that they can vary in 4 primary ways. I promise you'll have to know this for the upcoming test. So in one way that they vary is the location of the carbonyl group. Remember, carbonyl represents the C double bond O. So you can either find it at the end of the molecule or in the middle. If it's at the end, it's an aldose, and if it's in the middle, it's a ketose. So notice all the sugars are ending with ose. Another way that they're structurally different is the number of carbons. For example, when we look at deoxyribose and ribose in DNA and RNA, notice that there was 5 carbons, so that's another name for it is a pentose. If there's 3 carbons, it's a triose, or if there's 6 carbons, like in glucose, then it's a hexose. Another way that monosaccharides differ is their spatial arrangement of their atoms. And so it's exactly where do you find the hydroxyl groups? In other words, the OH group. Is it above a carbon plane or is it below a carbon plane? And we'll see in future diagrams what I mean above and below the carbon plane. The last way that you can tell that they're different is whether sugars are linear or form ring structures. So sugars typically form rings in aqueous solutions, but you can find them as linear structures also. I love figure 5.1 because here you can see the difference in the placement of the carbonyl group. Notice an aldose has the carbonyl group at the end, where ketose has it in the middle. This looks like a beautiful test question. In the next figure, figure 5.2, we see the difference between glucose and galactose. Notice that both glucose and galactose have 6 carbons as a backbone. So what's the difference between the two? If you look at the 4th carbon, glucose has the hydroxyl group above carbon 4 or above the carbon plane. If you look at galactose and you look at the 4th carbon, you see the hydroxyl group OH below the 4th carbon or below the carbon plane. In figure 5.3, you see that you can see glucose as a linear form or as a ring structure. Now the interesting thing, if you look on the right hand side of the diagram, there's an alpha glucose and a beta glucose. So what's the difference between the two? If you look at carbon number 1, you see the hydroxyl group is below the carbon plane for the alpha glucose, but for beta glucose, notice that the hydroxyl group OH is above carbon 1 or above the carbon plane. So what distinguishes one monosaccharide from another? So let's go over this again. The location of the carbonyl group. Is it at the end or is it in the middle of a sugar? In other words, is it an aldose or a ketose? Number 2, the variation in carbon number. Is it a triose, a pentose, a hexose? And the different arrangement of the hydroxyl groups is the third way, hydroxyl representing the OH groups. And lastly, is it a linear form or does it form a ring? So all of these give clues to the different types of monosaccharides that exist. So where did monosaccharides come from? And how were they formed in the chemical evolution? Well, monosaccharides can be synthesized under early Earth conditions, and sugars have also been found on meteorites. So they've been found in space, so it could be that the first sugars actually rained down on early Earth. And that's sort of interesting to think that things that you could have rain that contain sugar. Okay, so how do we form a polysaccharide? Well, we have to connect the monosaccharides or the monomers together, the building blocks of sugar. Now, this chapter doesn't come out right and say it, but the default monosaccharide is always going to be my beloved glucose, C6H12O6. So how do we start forming different sugars? Well, if you link two monomers together, you're going to get a disaccharide. And di, the prefix di means two, and saccharide means sugar. It says when sugars are linked together through a condensation reaction, and they're linked by two hydroxyl groups, which we're going to see in the upcoming figure. So when this happens, that linkage is called a glycosidic linkage. And this is really important, and I promise you this will also be a test question. Sugars are connected by glycosidic linkages. So they're formed by condensation reactions, but they're broken by hydrolysis reactions. So there's two types of glycosidic linkages, and these are the alpha 1,4 glycosidic linkage or the beta 1,4 glycosidic linkage. At first, when you look at these linkages, it looks sort of like a daunting task to know the difference between the two and actually what they really mean. But once we look at a figure, you're going to see, oh, this is very easy to understand. So the 1,4 represent carbon 1 of one monosaccharide with carbon 4 of another monosaccharide. And this is where the glycosidic linkage takes place. And we'll see this in just a second. So it says here C1 hydroxyl groups are on opposite sides of the plane of the glucose rings, depending on whether you're going to have an alpha or beta glycosidic linkage. I love figure 5.5 because it explains the difference between alpha and beta 1,4 glycosidic linkages. On the left hand side you see alpha glucose actually binding to another alpha glucose. What's interesting is that the hydroxyl groups of carbon 1 and carbon 4 of the second sugar are below the carbon plane. And notice through dehydration synthesis, the removal of water, a glycosidic linkage is going to form. So OH and OH forms H2O, and what's left over is an oxygen. And lo and behold, when you look at the glycosidic linkage, there's an oxygen left that's binding the two monosaccharides together. Notice for an alpha 1,4 glycosidic linkage that the oxygen is below the carbon planes of both monosaccharides. Now let's look at the right hand of the diagram, and we see a beta glycosidic linkage, and this is between beta galactose and beta glucose. So notice the hydroxyl group for carbon 1 is above the hydrogen carbon plane, and for the second monosaccharide, the hydroxyl group is below the C4 or below the carbon plane. When a glycosidic linkage is formed through a condensation reaction, notice here that you see that oxygen in the glycosidic linkage ends up above the carbon plane, and this is going to form a beta glycosidic linkage. So what's the difference between the two, and why is it so important? We're going to find out that alpha 1,4 glycosidic linkages are very easy to break. On the other hand, beta 1,4 glycosidic linkages are very difficult to break. So cellulose or wood, we construct our homes and furniture out of it, and you can imagine it's made out of beta 1,4 glycosidic linkages. But on the other hand, eating starch from potato, it's mainly made of alpha 1,4 glycosidic linkages. Okay, so that's the first thing that we'll talk about is starch. So what is starch? Starch is a polysaccharide. So polysaccharide means it's made out of many saccharides or many monomers of monosaccharides. So it's composed of alpha glucose monomers. And so the linkages are alpha 1,4 glycosidic linkages. You can also have alpha 1,6 glycosidic linkages so that you know that the second monosaccharide is linked at the C6 carbon. So it says here branches may occur every 30 monomers. So notice starch isn't very branched. It's almost just a long chain of glucose. On the other hand, glycogen, which is the storage of sugar in animals, is stored in liver and muscle cells. But notice here that branching occurs 1 out of every 10 monomers. So glycogen is much more branched than the starch is that is found in plants. I promise you this will be another test question. So starch is found in plants, and glycogen, another polysaccharide, is found in animals. Another big difference between the two, starch doesn't have very much branching, but glycogen does on the other hand. Another type of sugar is cellulose. Notice that cellulose, instead of an alpha, it now has beta 1,4 glycosidic linkages. That means it says every other glucose is flipped around so that the oxygen in the glycosidic linkage is above the carbon plane. And it says cellulose is so strong, it's such a structural polymer that it forms a protective layer around each of the plant cells. And this is what actually forms the cell wall. So now you know that cell walls in plants are actually made out of sugar, and that sugar is cellulose. It's so strong because of the beta 1,4 glycosidic linkages. Another type of sugar is chitin. And chitin is another type of polysaccharide, and it's found in fungi and it's found in animals. Another way of saying it is, it gives the crunch when you step on a cockroach. So the exoskeletons of insects or of crustaceans is actually formed from a sugar, and this sugar is called chitin. Chitin is also formed with beta 1,4 glycosidic linkages. That means every other monomer is flipped, so that when a glycosidic linkage is formed, that oxygen is found above the carbon plane. Another type of sugar is peptidoglycan, and this one is also a structural polymer, and it's so strong that it protects the cells of bacteria. And so this is why it's so important to take antibiotics over a period of time, because most antibiotics interfere or disrupt with cell wall formation, and it too is made from beta 1,4 glycosidic linkages. Table 5.1 is a great summary table because it goes over all the polysaccharides that we've just discussed. The first two, starch and glycogen, are made out of alpha 1,4 glycosidic linkages. When you think about it, does that make sense? These are linkages that can be easily broken. Do we want linkages to be easily broken? And the answer is yes. We eat a lot of foods that are made from starch. Notice that if you look at the molecule, it's a huge polymer or macromolecule, and notice that there's very little branching for starch. But if you look at glycogen, which is immediately below, you'll see that there's a lot more branching. Okay, so glycogen is found in animals and starch is found in plants. Then you look at cellulose, chitin, and peptidoglycan, the last three sugars, and notice how they look like strong fencing material, like a fabric of steel. And you can tell here that this is really a very strong structure, and not only that, the linkages are beta-1,4 glycosidic linkages. These are linkages that are very hard to break. A lot of work has been done analyzing the importance of each of the four macromolecules, and the truth is polysaccharides are very important, but they likely played little or no role in the origin of life. And one of the reasons is actually polymerization of monosaccharides requires an enzyme to actually carry out the reaction. So proteins are needed to actually build polysaccharides. They cannot catalyze their own reactions. They're reliant on enzymes. And the last thing is polysaccharides don't carry any information in the molecules themselves for information required for reproduction. There's no information that is passed from one generation to the next through carbohydrates. So, if they weren't important for the chemical evolution, then why are we studying them? Because they are very important. First of all, they're diverse, but why we study them is they're precursors to other molecules, such as nucleotides and amino acids that build nucleic acids and proteins. They're also fibrous structural materials. They're important for cell identity. And lastly, they're used for chemical energy. In fact, every time I eat a candy bar, I rationalize to myself that's the reason why I have to eat it, is because I need that chemical energy. Okay, so what do we mean by structural support? So cellulose in wood, chitin in insects, and peptidoglycan in the cell wall of bacteria, they form these long strands that are bonded on the sides and all around them with beta-1,4 glycosidic linkages. And keep in mind that these linkages are very hard to hydrolyze or to break. And so therefore they're very important for structure and they're very important for protection. So oftentimes when people say that they're eating a sugar-free diet, the truth is that we do need sugar in our diet, but likely in the form of fiber, right? Dietary fiber, which is a form of carbohydrate. Carbohydrates are also important for cell identity. So a lot of the proteins embedded in the cell membrane are actually glycoproteins. There's also such a thing called glycolipids. So if we look at both of these words, the prefix glyco refers to sugar. And when I think of glyco, I always think of glucose. So every time I see those words, I know it means sugar proteins and sugar lipids. So they're used for cell-to-cell recognition and cell-to-cell signaling. In other words, isn't it wonderful that our bodies know through glycoproteins that eyeball cells stay with eyeball cells and liver cells stay with liver cells? And this is through cell-to-cell recognition and also cell-to-cell signaling. In figure 5.6, this is a cell membrane, and we see that proteins are embedded in that cell membrane. But then you see all of these red rings, and these red rings almost look like antennae, right, popping out from the cell. They look like they're carrying out or finding out information, and that's exactly what they're doing. So those are polysaccharides that are attached to proteins, or in other words, they're glycoproteins. There's an experiment in your book that you can take a look at, but what they did is they actually took apart a glycoprotein. And what they did is some sperm only had the sugar part of a glycoprotein, whereas other sperm only had the protein part. And then they wanted to know what effect it would be on having sperm attached to eggs. And what they found is that the sugar was way more critical than the protein for attachment of sperm to the egg. So please take a look at this case study that's in your book. Carbohydrates and energy storage. So carbohydrates actually store chemical energy. When I sing to my candy bar before I eat it, and I say that you are my sunshine, my only sunshine, do I literally mean that? And the answer is, heck yes, I do. Photosynthesis means light making, and it's light making sugar. So notice that photosynthesis has 3 ingredients. CO2, water, and sunlight makes delicious sugar, and then oxygen is the byproduct. So that's really important. The ultimate source of energy on this beautiful world of ours is from the sun. And the only way that that energy can be used in a form on Earth is through plants, bacteria, and algae that carry out photosynthesis, light making sugar. Carbohydrates have more energy than CO2. Now, why is that? Well, because carbohydrates are larger molecules and therefore they have more bonds. Now, they have several types of bonds. They can have carbon-oxygen bonds, but they can also have carbon-hydrogen and carbon-carbon bonds. So, which of these bonds supply the most energy? Well, I know you guys already know the answer. It's those bonds that are non-polar covalent bonds, those that have a net neutral charge. In other words, they're sharing their electrons equally. So that's going to happen between carbon-hydrogen and carbon-carbon bonds. So what about carbon and oxygen? You already know that oxygen is electronegative, and that means it's going to attract electrons towards the oxygen atom. And when it does that, it's going to form polar covalent bonds. Electrons are not shared equally, so they will not store as much energy as the carbon-hydrogen and the carbon-carbon bonds. Now, if we compare fats to sugars, actually fats hold more energy than carbohydrates. And the reason is because fats are mainly made of carbon-carbon and carbon-hydrogen bonds. I promise you this will be a test question. So in figure 5.8, you can see the difference between carbohydrate and fatty acid bonds. Notice that when you look at part C, a fatty acid, which is a fat, you'll see that for most of the molecule, except for the tail end, is that you have non-polar covalent bonds. You see that electrons are shared equally between carbon and hydrogen and between carbon and carbon atoms. But if you look at carbohydrates, on one side of the carbon backbone, you'll see hydroxyl groups, that OH. And every time you see an OH, you know that the electrons are going to move to the side of the oxygen because oxygen is electronegative. So those bonds are weaker than the bonds formed between carbon and carbon and hydrogen. So therefore, which one has more energy molecule per molecule? A fatty acid has more energy than a carbohydrate. Okay, so what are some important enzymes that hydrolyze polysaccharides to release glucose? So let's go back and let's take a look at the word hydrolyze. What does hydrolyze mean? It means to break down, and in this case we're breaking down polysaccharides, a long chain of glucoses chained together with the glycosidic linkages. So this is done through hydrolysis. So what are some of the enzymes to do that? So one important enzyme is phosphorylase. Phosphorylase is the enzyme that hydrolyzes glycogen. So this is what breaks those glycosidic linkages. Starch is hydrolyzed by an enzyme called amylase, and amylase is found in your saliva. So if you keep on chewing on a cracker and you chew and chew and chew, after a while, instead of tasting salty, it's actually going to start tasting sweet. And the reason for this is because you're breaking the alpha 1,4-glycosidic linkages, and when you get to monosaccharides and disaccharides, they're tasting very sweet. So when cells need energy, it needs glucose, and that's the breakdown of glycogen and starch. And this energy is converted from glucose into ATP. So there's a whole chapter on cellular respiration, and cellular respiration is taking a sugar in the presence of oxygen and breaking it down from a 6-carbon molecule all the way down to CO2, which is a 1-carbon molecule, to actually convert energy to ATP. The energy in ATP is used to drive reactions, and those reactions are endergonic reactions, meaning that they require energy. Here we see all different forms of where we can get or we can find sugars. So we can find simple sugars like fructose in fruit, and we can find long chains of sugar in bread, maybe carrots and rice and grains. And then cellulose is a sugar that's found in wood, and now we've gone from alpha to beta 1,4-glycosidic leakages, and then there we see a lobster, which indicates chitin, which is a very strong structural molecule.